Hello and welcome everybody to a new U of Care podcast. My name is Oliver Grundmann and I'm happy to talk today to Dr. Brandon Warren. Dr. Warren, do you mind introducing yourself?
Sure. Yeah, I'm happy to. My name is Brandon Warren. I'm a assistant professor in the College of Pharmacy in the Department of Pharmacodynamics. I'm relatively new at the University of Florida. I've only been here a few years now. I started in 2018. But I'm excited to be setting up my lab and really starting to do my own research. So I study drug addiction in the context of learning and memory. So I look at cocaine addiction. I train rats to take cocaine, and then I try to get them to forget how much they like taking cocaine. Essentially, my research looks at learning and memory. I use the hypothesis that memories are stored in the brain in these patterns of neurons called neuronal ensembles, right? So we stick this hypothesis that neuronal ensembles control learning and memory, and they control these learned behaviors, and drug addiction is one of these learned behaviors.
That's an interesting field of research as we explore our brain further, and that has been actually an initiative I believe that was initiated under a former President Barack Obama, kind of like mapping the brain, if I remember correctly. And how do you get into this field initially when in the field of memory, and what got you interested into specifically then cocaine?
Yeah, right. So first of all, what got me interested in addiction research in general, I've always been interested in drugs of abuse. I've always been fascinated by the idea that these little molecules can so fundamentally change personality, motivations, and all of these things with the brain. And again, people's personalities can shift totally because of these little molecules. So I've always been interested in that. When I first started my research as a graduate student, I was studying the effect of drugs during early life. So we would give, methylphenidate, which is Ritalin or Prozac, to very young rats, and see what would happen to those animals when they grew up. So I got interested in learning and memory because I started doing another model where I was training mice to get socially defeated. I was not training them to get socially defeated so much as I was socially defeating them, which is a model where you put a mouse into the home cage of a larger, more aggressive mouse, and that mouse gets repeatedly confronted and overpowered, and eventually, he gets a depression-like behavior. So that's what I had become interested in this idea that depression-like behavior was sort of a learned behavior, right? The mouse wasn't depressed until we put him into this environment where he learned, that he did not like to get beaten up, basically. And then he would adopt those depression-like behaviors. So I started looking at the neuronal ensembles, not necessarily the neuronal ensembles. I was looking at the activation, the neuronal activation that was associated with that learning event. And that got me really interested in looking into neuronal ensembles, right? Trying to find the tools that we could use to manipulate those neurons. And the reason being is that most of the neuroscience research that has happened over the past 50 years or so has been looking at brain regions, right? So we do these manipulations to a whole brain region, but that's not really the way the brain works. The brain is made up of neurons. These are the subunit of the brain. They are activated by different events and stimuli, but the way that the brain really works is that some subset of neurons within a brain region are the ones that are responsible for mediating a behavior. It's not the entire brain region, right? It is some subset of those neurons. So the tricky part is, how do you get at those neurons? And I found a population of these neurons that were activated, by social defeat, and wanted to figure out how I could manipulate them. So there are really only a few labs in the world trying to target those at the time, trying to target those activated neurons. So Bruce Hope at the National Institute on Drug Abuse was one of those labs. So he was developing some really nice tools to work with the specific neurons. So again, it is about 4% of the brain that is being activated by cocaine or opioids. So it's a really small population. So how do you manipulate just those 4% of neurons that are being activated without affecting the other 94%? So I joined Bruce's lab in order to figure out how to do that. And he was using immediate early genes like FOS. So FOS is an immediate early gene that is expressed only when the neuron is strongly activated. So around 90 minutes after strong neural activation, the neurons express FOS. Right, so he was able to develop some really nice tools based on that. So he had a couple of transgenic rats at the time, a FOS-LAC-Z rat. This is a rat that expresses beta-galactosidase under the control of the FOS promoter. So whenever the neuron is strongly activated, they express this beta-galactosidase, which doesn't do much on its own. But once we inject another drug called DONO2, the DONO2 causes those activated neurons to die, basically. So what this does is allow us to selectively lesion only the 4% of neurons that are activated. And this really was fascinating to me, and it got me started on drug addiction, again, because that's what they were doing. They were at the National Institute on Drug Abuse. They were doing self-administration models. And to be honest with you, I didn't want anything to do with self-administration going into that, because I had heard sort of the horror stories about how you have to be there, night and day, training the rats, putting them in the box, over the weekends. Which is true. It's all true. I mean, it is a lot of work to get rats to self-administer drugs. But I also think it is, extremely rewarding. And this is one of the things that I learned when I got to NIDA, was how much I liked doing it. Because, before I'd been doing these sort of assays where we're looking at depression-like behaviors, the force swim test, the elevated plus maze, novelty-induced hypophagia, these kinds of tests. And it's really not as clear cut, right? You measure the amount of time they're immobile or the amount of time they're in one part of the box versus the other. And it's a lot more challenging to interpret that than just putting the rat into the box and letting him press a lever, right? If he's pressing the lever, that he's pressing the lever to get the drug. There's no real other interpretation. He's pressing the lever because he wants to get the drug. So I loved that. I loved how clean cut it was. The rats, when they train to press for cocaine, they press for cocaine. They will press hundreds of times. It was beautiful to watch. It's not, a sort of there's nothing ambiguous about it. They press the lever, they want the cocaine. So I really liked that, and that was really rewarding. And it's easy to test too. That's another thing that I really liked. You can manipulate these neural ensembles, you can manipulate parts of the brain, and you can see whether they press the lever more or less to get the cocaine, whether they want the cocaine more or less, if they remember pressing the lever for the cocaine. So that's how I got started. And I really love doing it. So it's what I still do in the lab. I still am starting to do some of the stress-related research, because I think they're both, one of the things that I think is fascinating is that both, post-traumatic stress disorder and drug addiction are both learned disorders, right? You can sort of argue they are disorders that are learned, because, you're not a drug addict until you have that experience with the drug and you learn that you like it so much, and then you become, if things progress to drug addiction, you become a drug addict, or you get substance use disorder. Now, with post-traumatic stress disorder, the same is true, right? You don't have post-traumatic stress disorder until you have that initial event where you are subjected to the stress and you learn, how stressful it is. So that's one of the things that I've been really interested in. And both of those diseases don't have really good treatment. There's replacement therapy for opioid use disorder, but really there are not any good pharmacotherapeutics for either of those disorders. And one of the things that has been my goal is to try to figure out what processes might be overlapping between these two types of learning, because, again, I think they are learned disorders. So maybe there is some dysregulation of gene expression. There's some dysregulation of the learning process that amplifies the learning that's taking place in either of these disorders. So if we can find out what that is, maybe we can, find a pharmacotherapeutic that can treat, craving and relapse in cocaine use disorder, or, the post-traumatic stress disorder as well. So that's been my goal over the past few years, is to try to find out, how can we find out, what changes to gene expression or protein phosphorylation or other things are happening specifically within that neuronal ensemble. Because again, it is only 4% of neurons that we think are mediating these behaviors. So how do we find out what is happening just in those neurons? And that's been a real challenge.
So when you specifically say training and basically a learned behavior of substance use disorder, and you see this overlap with PTSD, have you actually seen that the same neuronal ensembles are being activated by PTSD as well as you have seen by substance use disorders as well?
Right. So I think, I have not specifically looked, but other people have, right? Other people have looked at, the population of neurons that are activated by cocaine seeking versus the population of neurons that are activated by, an aversive event, right? So again, I have not done it, but other people have, and it is different populations. So this is one thing that is fascinating about the neural ensemble hypothesis, right? It is not a set set of neurons. There's not a population of neurons that are just opioid neurons, I mean, sure, there are in some parts of the brain, there are neurons that express opioid receptors, but they're not a population of neurons that are, these are morphine neurons and these are aversion neurons. So the neural ensemble is actually selected. It is a sort of a random population of neurons that are primed to be selected on a certain day. And then once they are selected by the reinforcers and by the stimuli associated with those reinforcers, they're sort of co-activated by the stimuli and reinforcers. And after they are repeatedly activated, they wire together, right? So they start to undergo these synaptic changes where they are undergoing restructuring and connecting to each other. So there's this saying, it is a Hebbian learning is what I'm sort of talking about now. And there's this saying that cells that fire together wire together, which is really true. So once these cells are all being co-activated at the same time over and over again, as the rats are, or humans too, as the learning is taking place, as these stimuli and the reinforcers are repeatedly being paired, again, the cells wire together and they form this ensemble. So the ensemble is this pattern of neurons that are all, wired together and encode this learned association. Right. So again, it is a random population of neurons. It's just, the cells that are activated on that day and reactivated, and many things go into it. This is another big challenge with this hypothesis is, we don't know which neurons are going to become the ensemble until they have become the ensemble. Right. There's several lines of research to try to figure out which neurons are going to be selected and maybe even force neurons to be selected for the ensemble. But it's really tough to know which population of neurons is going to become the neuronal ensemble beforehand.
So there seems to be, though, the underlying similarity seems to be a stress exposure, right? Because both, no matter if it's a substance use disorder or if it's when we talk about depression or we talk about anxiety, it's kind of an initial stress exposure that the body faces. Could that be a kind of an underlying theory that kind of causes this surge or this kind of formation of this neurological ensemble?
Yeah, that's a really good point, and I had not considered how sort of the physiological response on when they are first taking cocaine or something like that could play into the subsequent development of use disorder or drug addiction. But I think that's a good point. And maybe, as you point out, it could be that, and to be honest, I'm not an expert. I'm not sure. I'm an expert on some things. I'm not an expert on what happens in humans when they first take a drug. So I don't know exactly whether there are individual differences, which is, was I think what you are pointing out. Are there individual differences in the initial response to drugs that would then make them more susceptible to developing drug addiction? It's true. It's definitely true for post-traumatic stress that happens. I'm not sure for drug addiction, but it is something that I've been very interested in. Not necessarily. And this is what I think is interesting that you're pointing out, that maybe the stress response could be modulating their experience on the first day. But the first experience is something that I've been very interested in because I want to know how these ensembles are being selected, right? And one of the things that we've been doing in the lab recently, and it's something that I've been writing grants on, is to look at how neuronal ensembles are forming in that very first exposure, right? And are rats more or less susceptible to, subsequently train to take cocaine, or do they take more cocaine or less cocaine after that initial exposure and their response during it? So we've been looking at the rat's ability to acquire cocaine and then looking at other behaviors and trying to correlate them to see if, are there underlying differences in the rats that cause them to become more susceptible to train for cocaine seeking versus rats that don't. Because one thing that, in the literature with rodents and in humans too, I mean, humans, not everyone that takes cocaine becomes develops cocaine use disorder, right? It's a small percentage of people. And the same is true for rats. You can get them to train eventually, but, there's always a subset of rats that will not take cocaine, and there's a subset of rats that take cocaine more than everybody else. So one of the issues with cocaine self-administration in rodent models in general is that, it's not a really good model. I mean, it's probably the best model that we have of human substance use disorder, but it's not the best model because they're in a box where they can do nothing else, right? They're in a box, they have no access to anything else to do. They can't choose to do anything but press the lever. So they press the lever, they take drugs. And I think just about anybody who's put into a box with just a lever for, a few hours is probably going to press the lever, and maybe they will keep doing it if it feels good and they're getting an infusion of cocaine. So I don't know that is the best model. But, one of the things that we can do as well is if you give them a choice, right? Rats will generally choose food over cocaine, right? So if you give them a discrete choice, they will press the food lever, except for a subset of them. So that's the subset that maybe we should be the most interested in, right? They are the ones that are, developing this disorder. They're the ones that are choosing the cocaine over other positive reinforcers. But yeah, so that's one of the things that I've been interested in. How do we get from point A, where there is no ensemble, to an ensemble? And, are the rats more or less susceptible? Are there individual differences in susceptibility to acquire cocaine seeking? And if there are they based on, activation of the ensemble?
That is fascinating. And I think expanding on this, because you are in the College of Pharmacy, the same college as I am. So where do you see the major implications for your research, both related to substance use disorders, as well as you are looking at PTSD and some of the similarities to cocaine use disorder or stimulant use disorder, and for the future of addiction research?
Yeah. So I think, again, one of the big problems with treating substance use and PTSD is that there really are not great pharmacotherapeutics, and the pharmacotherapeutics that we have for substance use are generally replacement therapies. And I think, a lot of that may be because they're targeting the wrong sort of structures. And in general, we are targeting whole brain regions, right? So if you're affecting, 100% of the neurons in a brain region, you're really getting at the 96% that are not part of the ensemble and are not mediating the behavior. So I think the problem is really targeting therapeutics to the site where they can have the most function, right? So this is what I've been trying to do. And it's been tough to find a good biomarker or a good gene expression pattern that is associated with drug addiction in these ensembles. It's what I'm doing now. So we are doing single cell RNA sequencing in neuronal ensembles associated with cocaine seeking in order to try to find gene changes that are taking place just within those cocaine ensembles, right? So those are the ones that are firing together. They're the ones that are wiring together. And they're the ones probably being most affected by cocaine. So there are a number of things that happen when cocaine is on board. It is a dopamine transporter blocker. There's sort of more dopamine in the system. So you get something as well called glutamate overflow, right? So these cells are firing more. There's more glutamate in the synapse where these within these synapses, especially in the prefrontal cortex, which is where I'm looking. But you have more glutamate in the synapse, so you get glutamate overflow, and then you get different changes in the actual synapse, right? You get more larger spine formation. You get more synaptic activity, I suppose, or more changes in the synaptic restructuring. So this is what I've been looking at. I want to see, can we find changes in genes associated with sort of that glutamate overflow and synaptic plasticity within these neuronal ensembles that we can then target a pharmacotherapeutic for, right? Because I think that is the key. I think one of the biggest problems with drug addiction is that you have first craving and relapse. These are things that come from learned associations. You get reactivation of the memory for cocaine, like a place that you took the cocaine, or just a picture of some cocaine, and then this is going to induce relapse, craving, and then ultimately relapse. So this, I think, is where I'm hoping to find a way to treat drug addiction, right? So if we can prevent craving, we can prevent that cocaine memory, that overwhelming cocaine memory that causes craving and then ultimately relapse. That's where I would like to find some pharmacotherapeutic that can block specifically that association, right? And the key is to not block all associations, right? You don't want to forget that you like to eat cake, right? You want to specifically forget that you like to take cocaine. And I think there are differences. So there are differences between natural reward memories and drug reward memories, because drug reward memories form these strong associations because of that glutamate overflow that I was talking about, and because of their effect on the dopamine. It causes these unnaturally strong synaptic changes that ultimately drive cocaine craving. I think the same is true for PTSD. I think these are ultimately unnaturally strong memory. Maybe not unnaturally in the sense that they're being sort of boosted by drugs, but at least dysregulated associations, right? So how are they being dysregulated, and can we reverse that dysregulation by specifically targeting the changes within the neuronal ensemble, right? Within the 4% of neurons that are being activated?
So would you regard that as an unwiring or rewiring of these particular neuronal ensembles that just have created an unhealthy, let's call it an unhealthy
right.
wiring or connection of neuronal ensembles?
Yeah, I mean, yeah. So exactly, I think that's probably the best way to put it, is sort of a unwiring. I would not say rewiring, because we're not trying to sort of retrain them. We're just trying to block that specific association, right? So if we can just decouple those neurons that are mediating that specific behavior, that's the ultimate goal, right? At least for my research. So that's what I want to find, is some therapeutic that will be able to specifically target those neurons. And I think there are many ways that we might be able to do that, but that's the goal.
Yeah. I mean, we basically try to do that with like cognitive behavioral therapy, right? By kind of retraining our own brains to do something different, to replace a current behavior with a new behavior, overcoming fears or depressive disorders, or even PTSD, or in addition to pharmacotherapy, doing restructuring our daily routines to replace it with something new. But obviously, if we can support that with a pharmacotherapy that is much more targeted than throwing something against the wall and see if it sticks, what we do with antidepressants at the moment, for example, what we do with anxiolytics at the moment, where we, like you said, we just hit the whole brain or whole brain regions very unselectively, and it affects basically all of the receptors without being targeted, and all of the neurons thereby. That would be, I mean, your work would have implications for a range of other disorders, thinking about depression or anxiety.
Yeah, and I think you've hit on something else, right? So once we understand how these synaptic changes are taking place, right, and we know how they are driving these unhealthy associations, we can call them unhealthy, but at least they are very strong associations. One of the things that we can do is sort of reverse engineer that, so that we can shift the balance, right? So now we can drive those sort of unnatural synaptic plasticity changes to drive those positive behaviors, right? So you could work it either way. You could either decrease the unhealthy associations, or you could increase the synaptic plasticity changes that are taking place for those healthy ones, and really shift the balance.
I think this is really exciting work, and I really look forward to seeing more publications and more work come out of your group, out of your work, and emerge that then translates into meaningful and new drugs, new medications that can be used in the future. So thank you very much for taking the time today, for explaining and sharing your work with us, not only for researchers, but also for the general public, because I think this is really important work and shows the breadth of what we do within UF Care and at UF in general. Thank you very much, Dr. Warren.
Oh, yeah. Thank you for having me. I'm excited to talk about my work.
Okay. This was a new podcast of the UF Care. For this and many other podcasts, please go to our website, and talk to you again. Stay safe, everybody. Thank you.